TMEM16A, or transmembrane 16A, is a member of a family of structurally related proteins. The TMEM16 family contains 10 closely related homologs which all span the cellular membrane of eukaryotes and facilitate the movement of polar and charged molecules across this cell membrane. Curiously, however, only two members of the TMEM16 protein family, TMEM16A and B, are monoatomic ion channels. The remaining TMEM16 family members all function as lipid scramblases, allowing phospholipids to move to alternating sides of a cell's lipid bilayer. This raises the question, how can such closely homologous proteins transport such different substrates? While both monoatomic ions as well as the heads of phospholipids are charged, phospholipids are much larger and have long hydrophobic tails. Previous structural analysis of lipid scramblase TMEM16 proteins reveal they consist of an N, a C, and a transmembrane domain, and exist as a homodimer. The transmembrane domain is made up of 10 alpha helices and forms a polar furrow or trench. This trench provides a hydrophilic groove for the polar head units of phospholipids to rest in, while their hydrophobic tails project outwards from the protein into the hydrophobic interior of the bilayer. This allows the phospholipids to move through the hydrophobic interior of the lipid bilayer. But how would this open trench, exposed to the hydrophobic membrane interior, allow for the transport of chloride ions? Proposed answers include the formation of a pore between two monomers, the rearrangement of secondary structural features to create a pore, and the formation of a pore where one side is made of immobilized phospholipid heads. The aim of the present study was to definitively resolve this question and determine how TMEM16A functions as a chloride ion channel. In order to determine this, cryo-electron microscopy was employed. HEC-293 cells were first transformed with a mouse homolog of TMEM16A. The protein was then purified into detergent bilayers and samples were flash frozen with liquid ethylene. Cryo-EM was then used to elucidate the structure of TMEM16A to a resolution of 6.6 angstrom. Poorly defined regions of electron density were observed around the exterior of each side of the detergent bilayer. However, the transmembrane region of the protein was well resolved and revealed conformational differences to other TMEM16 family members. Crucially, 5 alpha helices that form an open hydrophilic groove in most TMEM16 proteins had moved together to form an enclosed pore, shielded from the hydrophobic interior of a lipid bilayer. Additionally, dilation of the pore at its intra- and extracellular sides resulted in a conical entrance for incoming ions. In order to further investigate unique aspects of this chloride-transporting TMEM16 family member, electrophysiology experiments were conducted. Patch clamp recordings were taken of wild-type TMEM16A at positive and negative voltages, and the flow of chloride ions through the protein was documented. This was determined to be equal in both directions. Following this, several point mutations were made to residues located in the conical openings to the pore. Removing positively charged residues from the intracellular entrance to the pore resulted in hindered ion movement out of the cell. Reduced chloride ion flow was also observed when positively charged residues were removed from the extracellular entrance to the pore. Additionally, removing positively charged residues from the center of the pore lowered overall chloride ion movement. Combined, these results suggested that TMEM16A functions like many other monoatomic ion channels. Cryo-EM revealed a reorganization of the transmembrane domain from an open channel suitable for lipid transport to an enclosed, hydrophilic, and positively charged channel suitable for chloride ions. This channel is similar to those seen in many potassium, sodium, and calcium channels and provides an ideal environment for charged monoatomic ions to flow through. Additionally, these data showed dilated openings to the pore which were speculated to allow for ions to more easily enter. As well as this, the slowly tapering entrance allows incoming chloride ions to lose their hydration shells, this a necessary step before squeezing through the narrow pore. The importance of these dilated openings and their chemical composition was also demonstrated when point mutations to residues around them resulted in reduced chloride ion transport. Additionally, electrophysiology experiments highlighted the highly optimized nature of TMEM16A in its role as a chloride ion channel. Many point mutations rectified or diminished chloride ion flow, detrimentally affecting its physiological role as a bidirectional chloride ion channel. This showed that TMEM16A elegantly fulfills its role as an ion channel, despite being the evolutionary descendant of a phospholipid flipase. To conclude, TMEM16A is a close homolog of many phospholipid scramblases, however it features a structurally modified transmembrane domain which is similar to many other ion channel proteins. Analysis by cryo-EM and electrophysiology experiments revealed 5 alpha helices reorganized to form an enclosed pore with a hydrophilic interior which allows chloride ions to pass through. The amino acid composition of this pore is specifically suited to facilitate chloride ion movement. Dilated openings to the pore also aid chloride